Okay, so here's our final little bit on the immune system itself. Here's a couple of little things to pick up, some fun things, I think, anyway, about how stuff works. So let's jump right into it. When we left off, we've been talking about antibodies, and antibodies are a little more, I guess, special than just calling them antibodies themselves. They do a lot of different things. They come in lots of different forms, okay? We're collectively going to call them immunoglobulins, not immunogoblins, but immunoglobulins, right? IG things. They're a class of different proteins. And the idea here is that they're going to bind to an antigen detected by a B cell. This is the whole idea, right? They come in 5 different flavors: IgM, IgA, IgD, IgG, and IgE. And they're all built around the same basic principle. There's a monomer, the basic structural unit, that is basically 4 protein chains. Okay, 2 of these are going to be longer, and we're going to call them heavy chains, and 2 of them are going to be shorter, we call them light chains. And they're going to form kind of this Y or T-shaped structure that we'll see on the next slide. The shorter chains are only on part of the monomer. The longer chains are on the entire thing. Where we have light and heavy chains together, this is going to be what we call the variable region. Okay, and this is what makes the antibody specific to one particular antigen. Right, this is where the antigen actually binds to the antibody. So here's what a monomer of our immunoglobulin looks like. Here we have the heavy chain. It's got a little kink in it, right? A little bend where we get the bottom of our Y and the arms of our Y coming off of it, one on each side here. And then the light chain is just going to be attached to the arms of our Y. The very, very end of each arm, where we have both heavy and light chain together, this is that variable region or the antigen binding site where we actually bind to the antigenic determinant. And so this is going to be the part that's different in different antibodies, right? All the rest of this nonsense is going to be the same. This is going to be the part that makes an antibody specific against one particular pathogen. As far as the different kinds, some interesting things about them. IgM is the biggest of all our antibodies. It's a pentamer. It is 5 of these guys stuck together. And this is usually the first antibody that we release. So the primary response, right? We release a lot of IgM. Okay. IgA is a monomer, so just one of these guys, right? Sometimes it's a dimer, which is 2. Okay, not infrequently, but it's not certainly the most common form of it. And we make this in mucus, right? So we talk about mucus being kind of a means to try to prevent pathogens from getting in. IgA is a part of that. IgD is also a monomer, and this is what we use on B cells as the B cell receptor. So when we talk about binding to a class 2 MHC on a B cell, this is what the IgD does. Our IgG, also a monomer, this is the secondary and late primary response. So we start off making IgM, and eventually we start replacing it with IgG. It's a little bit smaller, a little bit easier to kind of get around. And IgG will cross the placenta. So when we talk about naturally acquired artificial humoral immunity, this is what that's going to give us. So we get this from mom. And then finally, IgE, this is going to be what we see a lot in allergies and parasitic infections. And in turn, it will cause the release of our old friend histamine, which we all know and love from seasonal allergies and other things. Now, the beauty of these antibody types is that a B cell can actually make any one of these. It can decide that it's going to make IgM, and then it's going to make IgG later, right? In fact, this is what happens, right? So it doesn't matter if it's producing IgM or IgA or IgD, any of these guys, it can switch between it whenever it wants to. But once it makes one that's specific to a pathogen, it can't make a different kind, right? So IgG, IgD, IgM will always be specific to the same pathogen from the same B cell, from the same plasma cell. What's actually happening here? Well, the antibodies, again, go back and they opsonize, right? We've talked about this before, first video, right? We brought up the idea of opsonization. It's just a tag for cells from the innate immune system to come in and destroy things. But it also does 4 other things while it's doing that. So neutralization, okay, this is going to stop the activity of the pathogen in question, right? It's going to kind of coat the whole thing, and by kind of coating the whole thing, it's going to prevent it from making contact with other cells, from dividing. It's going to stop it from moving on with its life, which is a very, very useful thing to do. We're also going to agglutinate, and if you remember when we had those slides up of the blood types, right? When we had a reaction, those blood cells kind of clumped, right, and clustered. That's what agglutination is. And the idea behind agglutination is that we're going to kind of bring all of these things together in one clump or cluster so that it's easier to get rid of them, right? If you have, so I'll use my kid for example, if there are Legos strewn across the entire floor, it sucks to clean up. But if there's Legos in a little pile on the floor, it might be the same number of Legos, but it's easier to pick up. Okay, so this is what agglutination does for us. Precipitation is the same idea, except that it takes stuff out of solution. So if there are pathogens or other small things that are dissolved in the plasma, it brings them out of solution, again, kind of like agglutination, so we can get rid of them. And then, of course, the classical pathway of complement requires antibodies to serve as chaperones for us. Natural killer cells and cytotoxic T cells are going to operate the same way, but they're going to identify their targets in different ways. So if you remember, a natural killer cell looks for something that lacks an MHC class 1 complex, right? So anything that doesn't have one of those is going to be a target for a natural killer cell. It's also going to look for cells that have been opsonized, okay, by an antibody or by something else. Cytotoxic T cells, on the other hand, require an MHC class 1, okay, but they're only going to respond if there's a non-self antigen present. Right? This goes back to when we talked about the final step in maturation for the T cell. It could not respond to a self antigen, but it had to respond to a non-self one. Regardless of what the key is here, they're both going to destroy cells using the same process that we call a lethal hit. And a lethal hit involves the release of 2 different things. So the first part is that we're going to release these chemicals here called perforins, right, as in perforate. And the idea behind perforins is that they're going to act a lot like complement does. They're going to poke a bunch of holes in the cell membrane of the cell. Those holes then, okay, rather than just being stabbed a whole bunch of times and causing the cell to burst like we see with complement, those holes are going to be places where granzymes, the second thing that we release, pass through. And what granzymes do is they enter the cell, okay, and they basically cause it to commit suicide. We stimulate apoptosis in the target cell. And that's what the lethal hit is, and that's what we see with both NK and cytotoxic T cells. And these guys are constantly prowling. They're constantly circulating and looking around for abnormal cells, so virally infected cells, cancer cells, weird bacteria, whatever else, as a part of what we call the immune surveillance system. As far as other forms of T cells go, we've talked a little bit about helper T cells, right, in terms of what they do, activating B cells, activating CD8 cells. They also feed back onto the innate immune system. They can activate macrophages. They have a pretty profound impact on inflammation, you know, as so many other things do. But importantly here, without these guys, right, there's no immune response. Okay, without a helper T cell, we have no response whatsoever. Regulatory T cells are things we haven't talked about because they're a relatively small proportion of our T cells. A CD4 cell can make a helper T cell, right? It can also make a regulatory T cell. And really, mostly what these guys do, they help prevent autoimmune reactions to things. So they're small components, but really, really important for us. All right, final thing here, developmentally, immune system cells begin to develop as all blood cells do, first in the spleen and also in the liver, within that first 9-week period until bone marrow takes over a little bit later on. As you get old, your immune system function declines, right? For a variety of reasons, your thymus getting smaller, a handful of other things with that. What this means is that you are much more susceptible to disease, particularly immunodeficient or autoimmune disorders. We're seeing it right now with things like flu, with COVID-19, any of these sorts of things. Old people get hit the hardest, right? And eventually, because your immune surveillance system starts falling off, you're more likely to have a cancer. If you live long enough, you'll die of cancer, is kind of the end note on that. In the middle of these two things, where you're at right now, the biggest impact on your immune system are really activity of your nervous system and your diet. So I know it's hard right now, but stress, depression, grief, these are all things that are bad for a variety of reasons, but bad for the function of your immune system, which is something we're obviously really concerned with right now. So, do take time out for some self-care, take time out to kind of do some stress relief activities, because if we're going to get through this, we all need to be as healthy as we possibly can. Another aspect here is diet, in particular vitamin D. We live in the northern hemisphere. We're just coming out of winter. We're coming out of a period of limited sunshine. We're still in, as of today, period of limited sunshine. And so we are all going to be vitamin D deficient, okay? And a lack of vitamin D limits the ability for us to activate CD8 cells. So one thing you might imagine, right? Should I take a vitamin D supplement? Yes, you probably should. There's a lot of good research out there right now that vitamin D supplements are good for people who live in periods where limited sunshine exists. There's also a lot of good, so maybe you can stop taking it in the summer. There's also a lot of good evidence to suggest that there's a link between a mild, like a 25 microgram vitamin D supplement, and reduction in upper respiratory and lower respiratory diseases, things like influenza. If you're curious about this, talk to your pharmacist. Don't listen to me, but talk to somebody who's going to be a better at diagnosing what kind of dosage you might want or need. But it is something that has actually been shown. Unlike multivitamins, things like that, there is some evidence to suggest that this is helpful. On top of that, there is a link between vitamin D deficiency and MS. Whether this is causal or whether this is simply something that happens alongside it, we don't know. So I wouldn't go out and start talking about how vitamin D is going to prevent MS or whatever, but there is some link there. As to what it is, we're not entirely sure. Okay, so that's good for the immune system. We'll call it there. I will see you guys again on the next set of videos. We'll talk about the respiratory system. As always, if you have questions about any of this stuff, please feel free to comment, I guess, on the video itself if you want to, or send me an email, and we'll get it all squared away.